Well, hello, welcome to today's lecture by Dr. Jordon Beckler. I'm Dennis Hanasak, research professor here at Harbor Branch and host of the series. This is our last virtual lecture in our winter series. Of course, as you know, we just had spring. So for the next 3 months, April to June, we'll have what we're calling our spring series. And the only thing that's changing is that we will go to a once a month lecture. They'll still will be virtual for the spring. We'll kick the new lectures off on Wednesday, April 14th at 4 p.m. with a lecture entitled Technologies for Monitoring and Sustaining Our Oceans in the UN Ocean Decade. So first, how many of you actually knew that 2021 begins the United Nations Decade of Ocean Science, which seeks to reverse the cycle of decline in ocean health? I think you know that both ocean health and technology are two of our major research themes. You've heard much about them in this series. For this lecture, our speaker will be Donna Kosak, who works in Melbourne for L3 Harris Technologies. Donna is an ocean engineer with over 30 years of experience in supporting the design, development, and testing of various scientific and engineering projects, including computer vision, robotics, fiber optics, underwater imaging and communications, and ocean observing systems. So that's quite the gamut of tools for observing, monitoring, and understanding the oceans if we want to address ocean health. So this lecture, I think you'll see, will nicely continue the theme of our last couple of lectures in our winter series. So today's speaker is Dr. Jordon Beckler. Jordon directs the Geochemistry and Geochemical Sensing Lab here at Harbor Branch and is also a joint faculty member of the FAU Institute for Sensing and Embedded Network System Engineering. That's quite a mouthful, so we like to use the acronym, which is iSense, which is a lot easier to say. He's a geochemist. He likes to work, his expertise really is on sediments, and he received his bachelor's and Ph.D. degrees in earth and atmospheric science with a focus in chemical oceanography and a minor in inorganic chemistry from the Georgia Institute of Technology. Before beginning his position here in 2018, he was a Golf Research Program Early Career Fellow with the National Academies of Science, Engineering, and Medicine, and a staff scientist and program manager of the Ocean Technology Research Program at Mote Marine Lab over in Sarasota. So let's welcome Dr. Jordon Beckler to today's virtual podium for his lecture on ocean blue holes, a biogeochemical treasure trove. Jordon.
Hey, thanks, Dennis. Appreciate the intro. I presume that everyone can see my screen sharing. If not, please yell out. It is my pleasure to be here today talking to y'all about, perhaps the most exciting project of my career thus far. And the work is obviously a product of not just myself, but a ton of collaborators. I'm going to try to keep the research as straightforward as possible, but just sort of pronouncing biogeochemistry is difficult enough. So as you can imagine, it's somewhat difficult to try and present information in an ingestible format to folks that aren't geochemists, but I'm going to do my best. And I tried to minimize, the real nitty gritty science. I put in a lot of videos, so it should be a lot of fun. And so overall, I just want to thank my own lab group. So Matt, my student, and I'm going to get these acknowledgements out of the way in the front where they should be, front and foremost. Matt Quinnen, he's my student. Lynn is our lab coordinator. Andrew is a former student who worked on this project. Mason is a new student who will be joining us come May. He's already done some sample analysis on samples from this project. At Georgia Tech, we have my former PhD advisor, Dr. Martial Taillefer, who I still work closely with, and Nastasia, who is a friend and colleague, and she just started a position at NOAA, at a NOAA lab in California. She did a lot of the microbiological analysis. Over at Mote, we have Emily Hall and Jim Coulter and some other folks. and they've done a really nice job on the logistics around this project. Frank Stewart is a biologist at Montana State who's continuing the microbiology. Chris Smith at USGS, he is really looking to see if there's groundwater flow in these blue holes, which is super cool. Don Nuzio at Analytical Instrument Systems, he provides a lot of our technology. And finally, last but not least, the volunteer technical divers who have done a fantastic job in facilitating a lot of the work here. So Marty, David, and Drew, as well as some others. Oh, and I cannot forget Kristen at PBS. And I'm going to show quite a few of the videos that came from their Changing Seas episode. And then thanks to Marty as well, I'm going to show quite a few of his videos also. So thanks for bearing with me on that. So our current lab, as Dennis said, Geochemistry, Geochemical Sensing Lab. These are our current lab members. We're constantly changing. We're growing thanks to a recent project, which I'll talk about at the end of this talk, related to Lake Okeechobee algae blooms. Our lab is right here at the edge of this Lab 2 building. My office is right across the hall, so I get a really great view of the Indian River Lagoon. Our lab is nice and relatively tidy for a geochemistry lab, so I'm kind of a OCD neat freak when it comes to that stuff. Let me continue. So our lab, we primarily focus on what we call redox environments. So if you remember your Chemistry 101, we're talking oxidation and reduction chemistry. Sediments, marine sediments that are underlying water bodies are, perhaps the most iconic of these environments. Sediments are super dynamic and layered, right? So we use the term stratified. You can see the layers in these various sediments from these different environments. And that's primarily because of microbial activity. And the microbes, there's a whole interesting dynamic going on here. We call it diagenesis, and not many people even know what that word means, yet I've dedicated my entire career to it. And with diagenesis, it's important because basically the processes that happen in these top 10 centimeters or so govern how the sediments are going to interact with the water column above it. So take, for instance, the algae blooms in Lake Okeechobee, which are primarily attributed to sediment processes. And, there you go. You can just understand just how truly important this near surface sediment layer is. Sediments are, of course, at the heart of all different sorts of industries. And I spent a lot of work thinking about this slide and all of the ways in which sediments can interact with our world. The ones that we mostly focus on in our lab are human and ecosystem health, of course. Restoration of seagrass. Dennis Hanasak, who introduced me, him and I, we actually had a previous project about this. Food security, looking at sea vegetable aquaculture and sediments. Even using sediments to generate energy. So there's a lot of cool things that folks don't realize, a lot of cool roles that sediments play. So in pursuit of sediments, we have explored all over the world. As you can imagine, a lot of work in the Southeast, where I've spent the majority of my career. We've worked in the Eastern Tropical North Pacific. We've worked at hydrothermal vent fields, got to dive on the Alvin submarine. We worked in the Congo River deep sea fan, which was perhaps the most exciting month of my life on a French research vessel. We did some work in Puerto Rico. We actually do a lot of work in the Gulf of Mexico, surprising amount of work in the Gulf of Mexico, including not just in the blue holes, but also over in the dead zone where the Mississippi River outflows. And so our Florida projects, I'll talk today about the blue holes, which I'll also talk about their potential connection to Florida red tide, which we study. Lake Okeechobee and its primary conveyances, the Caloosahatchee River and the St. Lucie River. And then finally, we have quite a few projects in the Indian River Lagoon estuary itself. Now, I use this map in particular because, as you notice, there is the karst carbonate platform, right? So this, I believe, used to be above water 10,000 years ago in the last ice age. And so this was all above water. And so this was all receiving fresh rainfall, right? And that's going to really come in later. And I'm gonna play this PBS documentary to introduce the blue holes because they do a much better job than I ever could. And so here's your first video.
In Florida, reports of large gatherings of fish in the Gulf of Mexico piqued the interest of divers. They discovered an unlikely find beneath the fish. Deep, dark holes opening down into the seafloor.
You're swimming along on the bottom and it's very sandy. You'll see some fish go by, some random organisms around, but then you approach these holes and all of a sudden it's just booming with life.
These openings, commonly called blue holes, vary in size and depth. Some have only a small entrance that's 5 feet wide and opens up into a large room, while others extend down over 400 feet into the earth.
But the allure is, well, you wonder what's a little deeper and a little deeper. But pretty soon you're 150 feet or so and there's more to see. So you want to go deeper and deeper and find out what's going on.
These environments are so understudied that we don't even have the basic information to just characterize them.
And so as far as the timeline, these blue holes in the Gulf of Mexico specifically were being explored by technical divers, diving enthusiasts, way back, so for a few decades now. Now, I didn't learn about blue holes until 2016, and you can see I plotted my own general interest in blue holes, a real fancy quantitative plot there. And we had, when I was at Mote Marine, Jim Coulter actually collected a sediment core from Amberjack Hole, and I analyzed it in our lab, and we included that in a proposal that we submitted to the NOAA Ocean Exploration Research Program. I then left Mote and came to FAU. The proposal was then awarded, so we decided to keep it at Mote, so this way the logistics would end up being a little more fluid. We continued to test the technology until May 2019. We had our first cruise out to the Amberjack Hole. PBS accompanied us. They made a really awesome video. They joined us also for the wet season trip later on in that September. In July 2020, the video aired, the PBS video, and this also spikes with a Google search trend for interest in blue holes. So I think I can perhaps retire now that I've made a substantial contribution to science. And hopefully that's not the last, but that was just kind of cool to see. I did that the other day. In 2020, so just this past year during COVID, we went back to, we went to a different blue hole, the Green Banana Sink. Earlier this year, just in January, Anastasia led a paper in the ISME journal, which is the world's foremost microbial journal. So that was a pretty big high point for us. And then finally, to wrap things up for this grant, we will be going back out in May, back out for the Green Banana for the dry season sampling. And when I say, media circus, the PBS documentary really brought a lot of attention. So there had to be a few dozen articles, in the leading newspapers, national newspapers, ABC, BBC, international newspapers, I guess. So, it was a really exciting time to see our work being, all over the media like that. And so really though, with NOAA Ocean Exploration Research, they're interested in exploration, right? So science is allowed, but it's not supposed to be front and foremost. And so naturally, considering that the previous explorations of these blue holes were limited to these advanced technical diving enthusiasts and published in such thrilling magazines as Underwater Speleology back from 1994, it seemed like a perfect opportunity for us to focus a more targeted scientific effort and really undertake the first truly scientific exploration of these holes. So we're going to focus on exploring these holes, but we're going to explore the science. And that was kind of how we twisted it in a way, because you have to be creative when you're writing proposals. You have to twist things, right? And so these features, like I said, poorly known, great ability to capture public attention, therefore perfect for a NOAA OER. So we decided to focus on these two holes, Amberjack Hole and Green Banana Hole, both about 30 or 50 miles offshore of Sarasota. Initially, I said, do we really need these divers? I mean, you have remotely operated vehicles that can dive down in these holes. Do we really need divers, or is this just an excuse for the divers to go out and participate in science? And that was my initial thought until we actually got out there and I realized how challenging these holes are to work in. They're very narrow. They're full of silt, right? So if an ROV makes a mistake and kicks up a silt cloud, you won't be able to work in the hole for a couple of days. So I'm firmly a believer that the divers are truly necessary to work in these holes. And so how do the holes form? Here's a little background info, basically from two different processes. A sinkhole, right? Just like on land, imagine 10,000 years ago, sea level was a lot lower. You had the water, the groundwater of the fresh groundwater, which is of a low pH. It's acidic. It just eats away the karst bedrock. And finally, the cave, or you get a sinkhole that caves in, fills in, and then sea level rises. And now here you are stuck with a blue hole that's now underwater. So analogous just to how a sinkhole would form on land. On the other hand, you may have had fresh water flowing. Remember, you would have had fresh water near the blue hole as the continental shelf, as the West Florida shelf was exposed, right? And so this water could be discharging. This could have been even above water at the time. And again, this could have then filled in with water after sea level rose. And so a question is, especially with situation number one, do you still have fresh water flow? And initially we said, no way, it's 50 miles offshore. There's no way you could have any freshwater aquifer water coming out. And on the other hand, is the reverse true? Can you potentially be increasing the salinity of your freshwater aquifer, right? That's not necessarily a good thing. So these connections, this connectivity is obviously something of fundamental importance for our health on land as humans. I mentioned the red tide connection. So right now, you know, very few people, well, I'd say we don't really have a great grasp on what causes red tides. We know that there's quite a few explanations. We don't know which one is most important when. There's a lot of ideas, but not a huge amount of evidence to be out there. And so at least, you know, one observation is that the blooms do start offshore and at depth. And then eventually over time, takes a couple weeks or whatnot, the blooms make their way near shore and eventually upwell. And they expose, they rear their ugly head right near surface, or right near shore, sorry. Now imagine if blue holes are a direct conduit of nutrient-rich water delivering the nutrient-rich aquifer water literally directly to where we think these red tide blooms are starting. And I mean, it matches up. 50 miles offshore, that's where our blue holes are. So, you know, spatially, it makes sense. And so ultimately, we'd like to try to rank blue holes, or at least try to establish their quantitative importance as a potential nutrient source. So I modified this really nice figure from Cindy Howell et al. From 2014, where you can see near shore in estuary situations, sediment fluxes, atmospheric inputs, estuarine outflows can be important, but offshore, you're really limited to upwelling or some other complex processes related to atmospheric deposition potentially. But, you know, we're really after trying to figure this out. And only recently, really just this past week, did we finally get some good enough data where I think we could answer this question, but I just have to do the math now. Blue holes are ecological hotspots. They were really discovered by fishermen, we think. They're rocky outcrops in an otherwise barren environment. So naturally they provide shelter for fish, right? That's the number one explanation. But there is, in the early samples that our diving friends collected, there was elevated chlorophyll as well. And this suggests that for whatever reason, these blue holes may be a source of nutrients to the overlying water. And when you get the elevated chlorophyll, this is basically like seeing plants, right? It's like seeing the base of the food chain out at the blue hole. So for whatever reason, we think that these blue holes may be allowing the establishment of a base of a food chain and then somehow stimulating or perpetuating these the biodiversity in the area. And this is exactly what we're going to touch on later. You're going to see that essentially what we think is happening is that the blue holes are serving as funnels, right? They're funneling in this organic matter, this organic detritus, they're transforming it and they're spitting it back out and allowing primary production, or they're allowing the base of the food chain to basically exist. Here's a video from one of the divers, Marty Watson. This is from the Green Banana Sink. You can see all the fishing line there. So it is a dangerous place to dive because the divers could get wrapped up there. There's lionfish, shark, 3 sharks at one point. There's the scooter, he's swimming around. There's sea turtles, so all sorts of life there. So Jim Coulter, our collaborator over at Moat, who was actually diving these holes for a long time, he collected us a sediment core that we included the data for in the proposal. And the sediment core, notice this is from Amberjack hole. It's very silty, right? It's different than what you'd expect in the Gulf of Mexico and the surrounding the blue hole, which is basically just sandy and shelly and whatnot. And we used some of our instrumentation, we performed some electrochemical analysis, and we see, sure enough, that there is a bunch of hydrogen sulfide down in the top 200 millimeters, so 20 centimeters, top foot or so, loaded with hydrogen sulfide. And this to us suggests that the blue hole may be a source of organic matter deposition that's just being degraded in place, right? The sediments are essentially receiving a bunch of this organic detritus. Oxygen can't make it to the sediments fast enough. So organisms are breathing, but they're not using oxygen. They're using sulfate and they're generating sulfide in the process. When we see something like that, it seems to suggest to us that we have some sort of stratification. And these areas of stratification where we think that we may have oxygen on top, but no oxygen in the bottom, maybe we have warm water up top, but cold water down below, which would set up your density gradient, right? And prevent circulation, prevent the water from overturning. These type of sites are extremely high interest to people like myself. There's really only a few of these naturally redox stratified sites in the world. You have the Baltic and Black Seas, but they're far away and they come with their own geopolitical concerns. In California, you have a couple of basins just offshore. But then there's some on the East Coast, Chesapeake Bay, New England kettle ponds, but they all tend to have fresh water on top of salt water. And that's what causes them to have that density gradient. The blue holes, on the other hand, are purely marine. There's no fresh water on top. It's a purely seawater system. So it's a very unique environment. And to our knowledge, there's nothing like it in the East Coast of the United States. So it is indeed a scientific resource. And when I say all this talk about redox and whatnot, it's somewhat analogous to what we have on land here, or what I consider 2 end members, if you will, of, let's say, the Indian River Lagoon. In a healthy system, let's say a nice oxygenated system where you have a lot of life, a lot of seagrass bringing oxygen down to the sediments, oxidizing minerals and all this, you can manage to keep hydrogen sulfide at bay, right? All these oxygenated species, they sort of defend this more shallow sediment area from this toxic hydrogen sulfide. In a perturbed system where you don't have vegetation for whatever reason, low light penetration, so the plants can't survive, you get minimal oxygen delivery to the sediment, this hydrogen sulfide, which is toxic to most organisms, can creep upwards, even escape the sediments. In doing so, you also tend to generate more nutrients as well. The nutrients, they don't get trapped on these oxidized minerals in the sediments. There's nothing to hold them back, and you get additional nutrient flux in a process called eutrophication, where you can stimulate further algae blooms, and this sort of is a positive feedback that will perpetuate itself. And so, although a blue hole isn't necessarily a perturbed system in a way, because you get this minimal oxygenation, it's somewhat analogous to a perturbed system near land. And so that's kind of what we're thinking is happening at this point. And so what are we going to do when we go out there, we explore these blue holes? We're going to look at two of them, amberjack, green banana. We're going to go out in a wet and dry season to each one for about a week. We have 2 or 3 boats, 2 teams of tech divers, all sorts of guidelines that need to be set. Some of the boats are doubled or triple anchored. So, I mean, it's extremely complicated logistics. They're out there working a week or 2, even a month in advance, trying to get everything in place, the lines. If you think about it, we need scuba tanks set up for the decompression. The divers are down at 400 feet for only 15 minutes, but then they need 3 hours to compress. They're on rebreathers, right? So it's very different than your traditional scuba diving. So I already talked about our team, but this is basically who's responsible for what. And I'm going to talk about each of these things mostly individually. We're looking at we're trying to characterize the water column of the blue hole, both within and above the blue hole. So we're dropping a CTD rosette, a profiling package. We're deploying a benthic lander, which is an advanced piece of equipment, which we'll see. And that actually descends downward. In doing so, it collects a vertical profile, right? So we have instruments that are sampling as the lander descends, and then the lander sits there for a period of 24 hours or so, collects a time series of data, and then we get another profile on the way back up. We're also collecting water samples every 10 meters or so. The divers are helping out with that. And then while the lander is down there, it's directly measuring nutrient and hydrogen sulfide fluxes and oxygen fluxes for that matter. And then the divers are also collecting sediment cores for us, and you'll see how we process all of these. So here's what I mean. The divers, they look like they're going to outer space, right? They're on rebreathers with multiple air tanks. This boat is extremely crowded. There's no room to maneuver. It's actually, we made changes. We realized after the first couple trips, it was just too many people, right? So we had to really limit it. In terms of logistics, you'll just see the divers are basically, moving scuba tanks around, getting stuff in place, sliding scuba tanks along a line, just placing for decompression. So it's just a ton of work that goes into it. And what do I have here?
These holes and red tide
This
blooms,
is a long video.
experts say
It
more
introduced
research
a lot of
is needed.
sediment related.
I'm setting up everything to check the lander.
Another group of experts from Georgia Tech and FAU Harbor Branch custom designed a high-tech device called a benthic lander, which surveys the sediment and water at the bottom of the hole.
The benthic lander looks almost like the moon lander, something that we would put in space. It's also got a lot of instruments on it.
For this particular lander, we typically lower it on the seafloor from a crane on the ship.
In this case, with the hole being so narrow, it's a pretty small target. When you think about waves and things like that moving a ship around, the ship isn't really able to get the lander down there.
Since amberjack hole's diameter is only 75 feet, divers must guide the lander into the hole.
The lander weighs 400 or 500 pounds. So this thing sinks fast to the seafloor. We added flotation foam to it to make it completely neutrally buoyant so that the divers could physically maneuver it underwater and get it down into the hole.
That's something new that we had never tried before, so we're a little bit anxious about it. See if it's gonna work.
Technical divers can only stay at a depth of 300 feet for a short period of time, whereas the lander can continuously collect data over a 24-hour period. One component, called a benthic flux chamber, samples nutrients, carbon, and oxygen that transfer or flux in and out of the sediment.
Think about like taking a coffee can and inverting it on top of the sediments. Now you have a good little volume of water, and we have syringes that take samples from that little parcel of water every 4 hours, And they're sampling both inside the chamber and outside the chamber. Outside the chamber is sort of a reference, like a background. Inside the chamber tells us how much is actually coming from the sediments themselves.
And then also we have this electrochemical analyzer, we call it.
These electrodes actually allow us to monitor things like oxygen, sulfide, dissolved iron, dissolved manganese.
Once the experts determine the amount of nutrients and other elements accumulating inside the chamber, they can calculate the total amount of nutrients and other elements fluxing in and out of the sediment at the bottom of the hole. These measurements tell the scientists more about the microbial and chemical processes that occur in the sediments at different depths. To obtain additional information, Jim and volunteer technical divers also collect sediment cores.
So these are sediment samples. They came from the bottom of this particular hole at about 310 feet deep. And there's kind of what we call a debris pile on the bottom. This amount of sediment could represent, perhaps hundreds of years of sedimentation. Jordan will be looking at the chemistry of the sediment and see how it's functioning. And so we'll take it back and get it analyzed.
So we're gonna put the reference.
One core is used for further electrochemical analysis, while a second core is sectioned to determine how levels of nutrients and other elements change with depth.
So we're pushing the mud out using a piston, and we section it in small intervals. We then spin it in a centrifuge for 10 or 20 minutes. We separate mud on the bottom, water on top. We pour that water off, and then we filter it. We can use these pore water samples and actually conduct our analyses. Okay. And so we're in pursuit of basically these two different holes. And the reason we selected these holes, AJ is the first one we're going to visit. It is much larger. It's much wider. It's not really, it doesn't appear that way on this picture, but it's much wider. It's easier to access. So it's sort of a good training ground to go to Green Banana, which at times is only 30 feet wide or so. AJ, we think, used to be a sink, like a sinkhole. And Green Banana, we think, used to be a spring. Both of these sites, they have that debris pile down there, and they both extend beyond over 300 feet, right? Green Banana almost 400 feet deep.
Let's do both.
We collect water samples.
Scientists also collect water samples from the base of the hole up to the surface.
So that's using a Niskin bottle. That's how we collect the water samples above the hole and maybe down into just the shallow part of the hole. But then we need divers to actually collect the water samples for us down deep. I think this is Marty's footage. He's down there at almost close to 400 feet collecting a water sample. He did mix up a little more mud than desired. So they did go back and take another sample a couple days later because it's really critical that the water samples are, pretty pristine. So there's a lot of information that we have to coordinate with the divers, that we have to make sure that we tell them or ask of them. And so he just collected a Niskin sample from depth. You can see what the sediment looks like there. Now here is, sorry. I'm going to present the data from the water column now, and this is just a screenshot of the paper that Nastasia just led, and then it has all of us as authors. And like I said, it's in the world's premier microbial journal, so it's a good one to add to my CV. Thank you, Nastasia, if you're on. As far as the water column data goes, this is where it could get pretty complicated pretty quick, but essentially what we look at are these vertical profiles, and then on the x-axis, you actually see concentration scales or pH scales. And so here at this dotted line, this is the opening of the hole. This is the sea surface, and this is the base of the hole. This is all in meters. And first thing to note is just that your oxygen, it goes from being fully saturated. So the water is in equilibrium essentially with the atmosphere here in the above ocean. But as soon as you get to the mouth of the hole, it basically goes to near zero. We have some funky business going on down here where it goes back up. We're really not sure to this date what's causing that, but eventually we end up in a completely anoxic situation. Oops. We also, on the opposite side, we have hydrogen sulfide diffusing up from the sediments. It's transformed, it's oxidized to elemental sulfur. These are different redox states, which is then further oxidized to thiosulfate, which then interacts with iron, right? And we get the formation of this turbidity maximum. This is probably related to the elemental sulfur, these filamentous white organisms, or it's just the product of these organisms that are respiring. So these microbes, they don't require oxygen. They're essentially using these other alternative, this energy dense material essentially to provide them with energy. And so it's pretty unbelievable that we find all these different zones, all these different redox layers within this environment. And then finally, looking at nutrients, you see your phosphorus has a source from down below in the sediments. And finally, ammonium comes up as well. Ammonium's in blue. It comes up, but then it's oxidized to form nitrate, which is then either consumed at the surface by some process or just diffuses away. And so this is this full stratification layering. It's your stereotypical model data, right? This is what you would see in a textbook. So this blue hole, again, is a perfect scientific resource for this type of data. And finally, look at pH. It should be 8.1 if it's in seawater, but your pH is a lot lower in the blue hole. So the blue holes are perhaps a study site where you can look at future ocean conditions where you have, where you, in anticipation of you potentially having ocean acidification. The low sulfur, or I'm sorry, the low oxygen and high sulfide manifests itself down in amberjack hole as dead organisms. Nothing can live, we don't think. And you have 2 dead sawfish that we actually found down there. Gigantic fish that must have swum down there and essentially drowned underwater. FWC pulled them out a day or two later, and they said it was the worst smelling necropsy that they've ever performed. Now, the green banana sink or green banana spring is quite different, right? So whereas AJ, we saw the interface of where hydrogen sulfide met oxygen about 10 meters from the bottom, right here. And this is where we saw that filamentous elemental sulfur layer. In green banana, we don't see that because we have some level of oxygen throughout the entire water column, and we don't detect sulfide until we're in the sediments. So essentially, this interface has been moved to here, and indeed, we do detect white microbial mats right here at the sediment surface. So essentially, that's all that happened, is this interface was moved downwards. But how does that happen, right? How could you move this interface downward? It means that you have to be allowing oxygen in here somehow. We also saw some life down here. We saw a shellfish, or the divers saw a shellfish. We have it on video, which I'll show you. And the debris pile in green banana is a lot smaller than it is in AJ, suggesting that we have some resuspension and redistribution of these debris here. All of this seems to point to flow. So we think green banana has some sort of flow. The divers reported feeling the flow. We also have what we think is video evidence of the flow. Here's a couple of examples. Here's the diver. I'm going to show the shellfish. Watch closely. It happens fast. Right there. See that? That was a track in the mud where we think there was a shellfish that essentially just walked along the mud. Now that animal likely could not have survived if that was a purely sulfidic environment. We didn't see anything like that in amberjack. Now I want you to look for the particles. Look at these particles. Even when the diver stops, or especially when the diver stops, these particles are still moving. They're still flowing upwards. We think this is more it's just a ton. It's a big picture of a bunch of circumstantial evidence that we have that seems to suggest that you do have flow at this green banana sink. Now I'm going to switch gears a little bit. We're going to talk about the lander, and I just want to highlight how talented these divers are getting the lander deployed. This is quite a tight squeeze. There's the green banana hole. It's not very wide, what, 50 feet across? I guess this is the constriction. I think we're already midway down the hole at this point. And the divers have to very carefully maneuver their scooter or maneuver the lander using their scooter. So it just barely makes it in. You know, the hole is not much wider than the lander itself. Pretty wild. And then finally, this is my favorite. I'm going to not talk. I think the words touchdown as the lander touched down into the bottom of green banana. The divers, they're breathing helium, and so that's why they sound like Mickey Mouse, Daffy Duck, I don't know. So the lander gives us some really cool results. The lander's down there for 24 hours. Inside the benthic flux chamber, we should be collecting, it's in contact with the sediments, right? So any nutrients like phosphate or ammonium that are fluxing out of the sediments should accumulate in the chamber. And sure enough, the blue line, the pink line, they're accumulating in the chamber, but they're not accumulating outside the chamber, which is the dashed line, which is like a control sample. And so then we can actually take these rates and we can then determine a flux, and then we could figure out basically the quantity of nutrients that are coming out of the entire blue hole, or at least coming from the sediments of the blue hole into the water column of the blue hole. Now, the most exciting thing that we saw wasn't necessarily from the nutrient data, but was from a conductivity, temperature, chlorophyll sensor, and oxygen sensor that was mounted to the lander. You could see here during the daylight hours, your salinity is basically pretty constant, right? It's high, 36.89, something like that. It's high. And then all of a sudden, come 8:30 at night, your salinity drops rapidly and then fluctuates. What would cause your salinity to drop like that? Well, aquifer inputs. Now, it doesn't drop to zero, so we don't have salinity zero water flowing in, but we do have relatively fresher water flowing in. So you got to think the aquifer is like Swiss cheese, right? So we don't expect to have fully fresh water, but we have some brackish water, just really it's full seawater strength, but it has been diluted relative to what's already in the blue hole. And you may think, well, and is it related to tides or something? But as you'll see from the next slide, on a Monday overnight and then on a Thursday overnight, 3 days later, this blue and this orange line, 2 separate nights, 3 days apart, 24-hour cycle, your salinity basically like clockwork within 3 minutes of each other, both nights, it drops. So we think this is not so much related to the moon, but Chris Smith, the USGS, he suggested that this may be related to the land sea breeze cycle. And indeed, we're looking to see if we can directly connect it to that. So when this supposed or hypothetical aquifer water comes out, we also see massive increases in chlorophyll, decreases in oxygen. This all is consistent with it being water that is stuck down in the aquifer and being completely anaerobic. And then we also had some other really cool novel sensor on the lander made by our friends at Analytical Instrument Systems. We're really excited about this data, brand new test of some really new technology. We see hydrogen sulfide. Not very many sensors can detect that. So we see hydrogen sulfide only when this low salinity aquifer water is coming out. We even see thiosulfate as well, which is it's the product of sulfide oxidizing, reacting with oxygen to form that thiosulfate. Notice when these two are opposite. When you have sulfide, you don't have thiosulfate. And now this is what we look at Nastasia and myself, we drove ourselves crazy trying to understand, trying to really learn about sulfur cycling, sulfur redox cycling. Essentially, there's all these microbes that can do all these crazy tricks and extract energy from all these processes. And, you know, many of them are very poorly understood. And so this is what I mean when I say these dynamics can really help us further understand how these microbes are doing their trick. And these microbes may be very similar to microbes that lived on early Earth or may live on other planets. And that's, you know, one of the main drivers of looking at this research. Not to mention all these processes really control the nutrient transformations and thus, you know, may connect to things like algae blooms. So on the sediment course, we do what we call electrochemical profiling. We have an electrode that has here's a zoomed in tip. It's a little 100 micron, a little human hair diameter gold wire that we coat with mercury, like the silver mercury. The electrode penetrates the mud in very small increments. Over time, it takes a few hours. And at each depth, we stop and we run a series of voltammetric electrochemical scans. We apply a range of potentials, we monitor the current, and at each potential, not at each potential, but at certain potentials, if we see a spike in current, we can attribute it to a specific species like hydrogen sulfide, a specific chemical analyte. And then we could essentially convert those currents to concentrations and then construct a vertical profile of sulfide, of pH, of manganese, of all different types of analytes. So this provides us a really nice portrait, a picture of the processes that are happening. You saw in the picture, we developed a really cool technique. My student Matt just let a paper submitted to share this design. We had help from a Harbor Branch engineer in designing this system to process sediment cores really efficiently, and we do this in a bag that's filled with nitrogen gas. It's really a pain in the butt, but we have to do it because oxygen is our enemy, right? If we're trying to maintain redox speciation, we use nitrogen gas, which doesn't react with these oxygenated species. And so when we put it all together, we get the electrochemical profile, and then we also get the nutrient profiles from the pore waters. And you could see, for instance, your ammonium and your phosphate and even your hydrogen sulfide here, they all increase, right? They all increase over the top few centimeters. And when we see an increase like that, we know as geochemists, we know that there's a flux. We know that these nutrients are originating in the sediments and they're fluxing out. And we could calculate that rate and we can determine if blue holes are indeed a source of nutrients. And so I'm going to wrap up the blue holes right now, but basically three major findings, implications, whatever you want to call them. This funnel shape of the blue hole really helps attract life and sustain life. It's not just shelter. It's the fact that you have detritus, so dead fish and excrements and all sort of whatever you want to call it, coming down, sedimenting in the debris pile. It's transformed by microbes. It generates inorganic nutrients that can then flux back up. They're transformed all along the process by these different microbes living in these different zones. But then they end up fertilizing life at the top of the blue hole. And what does that life do? It attracts other life. And so it's like I call it an ecological engine. And I'm looking for a better term. I don't really, I keep thinking about this. Sometimes I try to dream about it. It's a physical process, very specific. And if you've ever seen the movie Animal House, I think it's kind of like the zit scene with John Belushi or Jim Belushi, I forget, but with the mashed potatoes. If you don't know what I'm talking about, just forget it. But anyways, there's obviously major implications for marine protected areas and habitat restoration if these really are not just habitats for life, but also just, but they're also places where life essentially begins. Then we should protect these areas. They're easily accessible redox stratified basins, and I want to explore this with a big proposal we're going to put in this summer. And finally, I really want to establish, are these nutrient sources for red tide? And we can do some back of the envelope math, and well, we've already done that math, and sure enough, it seems like, yes, the blue holes can indeed be a nutrient source for small blooms when they start in the Gulf of Mexico, but you really have to consider a whole bunch of factors before you can draw conclusions like that. And even trying to demonstrate something like that is super challenging. So that's, pretty far down the road. Last, I have 3 slides left. I just want to talk about our other big project that we have right now. Maybe I'll talk about it a year or 2 down the road. I don't know. But it's called the Harmful Algae Bloom Assessment of Lake Okeechobee, or HALO. It's a state-funded effort. And we are trying to understand what's causing the algae blooms in Lake Okeechobee. And the reason I'm really talking about it is because we borrowed some technology that we use from the blue holes. So we designed a smaller benthic lander that we're actually able to deploy off of our 28-foot Dusky at Harbor Branch. We actually just deployed it yesterday morning, Mason and I. And the lander, it has the benthic flux chambers, it has the electrode microprofiler on it, and we're going to be out there deploying it pretty much every month for the rest of the year. And our goal, again, is to try and directly measure the nutrient fluxes coming from the sediments. The project involves all sorts of people, including several in my lab that are funded basically 100% by this project. And so it's a big, it's a big activity at Harbor Branch. So I figured I'll shamelessly plug for it. And with that, I'm going to wrap up. And of course, I'll take questions, and Dennis is going to mediate them. And thanks just so much for your attendance. And again, thanks to all our collaborators.
Well, thanks, Jordan. I think that was really good. I can safely say for
Thank you.
everybody, those were super videos.
Mm-hmm.
I think videos is so effective to actually show what it's like.
Yeah, it's great to have those resources, I mean, heck, PBS made a documentary, right? Like, come on, how could I not show those? And then the divers are, of course, that's their craft. That's what they're doing. They're down there trying to collect solid videos so they could share them with their community.
Okay, so just remind everybody, we put the slide up there, but just go ahead. If you have any questions, go ahead and put it under the Q&A. And that would be a good thing. There's several already in the queue. Are you ready, Jordan?
Of course.
The first one's kind of cool. So
right.
How are these blue holes named? I mean, they have some kind of interesting names. So somebody would like to know, how do they get their names?
Well, it's only by the time I hear about the blue holes, it's all folklore, right? I've yet to name a blue hole. Maybe one day they'll name one after me. I don't know. But the green banana, I think there was fishermen who saw a green banana floating by. I don't know if they thought to conclude that it came from a submarine spring. I doubt it. That'd be quite the transit from the terrestrial aquifer for a banana to make it. I don't think that could happen. But amberjack, well, that one's aptly named. All you got to do is look at the videos. There are tens of thousands of amberjacks there. Some of the other ones was their megadome. That one's cool because it has only like a 5-foot entrance, and then it opens up to be a giant dome. And so it's the divers that name them, I believe.
Mm-hmm. Okay, so someday there's going to be a Beckler blue hole, you think, or?
I don't know. That's pretty lame. You can come up with something better than that.
Come up with something a little less lame, huh? I see a question from one of our colleagues, Bill Lauda, professor
Bill.
down in Boca.
Mm-hmm.
Has some similar interests, of course, that you do. He wants to know why chlorophyll in the supposed deep aquifer waters input at the bottom of green banana, ? Input of photic waters into a downwelling area into the aquifer that now upwells in green banana, ?
Yeah, I mean, that's an interesting thought that I hadn't really considered. that would be like a hydrothermal vent, I guess, where you have the convection of the water coming in from around the hydrothermal vents almost. But in this case, you're talking about on land, I guess, right? Theoretically, unless you do get some penetration around the blue holes in between land and the blue holes themselves of some relatively saline water. But honestly, Bill, I think it's just an issue with the sensors. I think the sensors are not specific for chlorophyll in that case. And I think you have some other organic matter that's just, reacting at those same wavelengths. That'd be my guess.
Okay, so
Or it could be degradation products too, right? You could just have perpetual degradation of phytoplankton and pigments and whatnot, but you're the pigment expert, so you could tell me if you're likely to get intact, or at least the photoreactive pigments coming via diagenetic breakdown of the phytoplankton.
I'd say possibly, but not too likely. It would be just totally off the cuff, off the top of my head. But I got another question. You got a lot of questions.
Sweet.
Is there any relationship between blue holes and submarine volcanoes?
Submarine volcanoes. When I'm thinking, when you say submarine volcanoes, I'm thinking like hydrothermal vent, or you have black smokers. In that case, no, because those are all fully convective marine systems where it's all just salt water going in. But then I think there's also some submarine volcanoes, like the ones over in Italy. There's some famous ones, but honestly, I'm not too keen on those systems. And so I would defer to someone else in this case. I don't know if there are any experts listening, but if we're talking about hydrothermal vents and black smokers, I'd say no. Some other possibilities I'm not aware of, maybe. Thanks for the question.
That sounds like a good choice. Now, one of our research professors here, John Reed, has asked to actually talk live. So I don't know if John, you can unmute, or Wyatt, can you unmute John Reed for us? John is certainly a good person to weigh in on this.
No kidding.
John, are you unmuted? It shows that you're muted, so I think you have to unmute yourself, John.
How's that?
Yeah.
Yeah, good.
Hey, Jordan, I really loved your talk.
Thank you, John.
I love this kind of exploratory research. It's so cool because you don't know what you're going to see. And I had a question about the banana hole. Is at the bottom where it appears there's fresh water coming out, or fresher water, did the divers actually see the reentrance, the hole going into the rock?
Yeah, so couple comments there. One is I love the exploratory research as long as you keep the science part hidden, and that's an inside joke. You know what I'm talking about. Don't advertise the science. The divers did dive down there, and we have the video of the corners of where the green banana, it does, I don't know what you'd call it, but it sort of widens out at the very bottom again, right? And but apparently it just shows the walls connecting with the seafloor, so it doesn't look like there's any visible openings. And a couple of divers did a full circle around the bottom of green banana, and it's not that big, it's only 30 feet wide, so it only takes a couple minutes to go all the way around. And there is no visible connection to go wherever, into the distant aquifer or whatever. And now you got to remember, though, is that when we sampled in person, it was daytime, and we didn't really necessarily see these major flows until the nighttime. So it may be that if we went back at night with flashlights and whatnot, and this is totally something that we're going to do, if we get funded again to go do this research, we're going to stay out there all night. And, we may just not have seen it because it doesn't happen to a huge extent during the day, or it might even reverse. We think perhaps the flow is reversing during the daytime, And then, you get outflow at night and reverse flow during the day. And it's going to be cool going back there this May when it's theoretically the dry season. Because last time we were out there in September, when we would expect to have some hydraulic head pressure pushing on the aquifer or pushing down on land, essentially, trying to force the water out. So the answer is no, I guess, but it's still somewhat inconclusive.
I wonder if you could take like a mini rover, a small ROV, and just kind of park it down there in a 24-hour period to see if you see the flow changing or something.
Yeah, I'm gonna bug some of our colleagues, like Laurent. I'm gonna bug him to try and get a ADCP active for the next trip. But we're sort of limited on space on the lander. But yeah, we definitely need some real-time sustained recording down there.
Now, as you know, I've published a couple papers on some deeper sinkholes. One is just seaward of where you were diving. It's called Naples Sinkhole, and it's 600 feet at the base. And then off the Florida Keys, there are half a dozen sinkholes, some going to 2,000 feet. And one of them is actually called the Jordon Sinkhole. Must be named after you, no doubt.
So they have one. He already has one named after him. He wants two.
I know.
But yeah, that one's cool. That one's a half a mile in diameter. It's gigantic.
Yeah, that's huge, So I'd be really interested in looking at the one that's closer to Green Banana, just because it's not as wide. I feel like the one that's a half a mile, you're not going to get the nice stratification that you would necessarily get.
Right.
Just as, too much room for vertical circulation and turbidity flows and whatnot, or little eddies, vertical eddies.
All right, thanks, John. We got a whole bunch more questions, so let's try to get more folks here. This one is on the technical diving side. Why were the divers breathing helium and not oxygen?
Well, so they're on rebreathers, so they're essentially they're scrubbing the CO2 from their airstream, and they're just slowly giving themselves a little bit of oxygen. So there's only a small oxygen tank at any given time, but then I think they also provide helium as well. And I think the helium may serve to displace the nitrogen, but this isn't my area of expertise, but I think that's why they have helium.
Okay, so what was the effect of the deep water horizon oil spill on blue holes? Did oil or other pollutants accumulate in the holes?
What's cool about that question is that there has been, to my knowledge, one other truly scientific study of blue holes, not in the Gulf of Mexico, but I think the great one in Belize or the Bahamas. Actually, there's been more than that because there's been a couple in China as well. Now, they were looking at the layering, the specific one that was funded by National Science Foundation. They were looking at the sediment layering as a proxy for like the last 10,000 years. They're looking to see if you can detect when there has been major hurricanes in the Atlantic, I believe, based on the accumulation of sediment down there in the blue holes. And so it'd be really cool to see if indeed there was some hydrocarbons stored down in the blue hole. That's a cool idea that I never thought about. We did, the last time we were out, collect some samples so that some colleagues here at Harbor Branch, Tracy Mintzer's lab, they're also biogeochemists, but they have a lot of projects looking at plastics. So we're going to see if you could see, the rise of plastics with regards to human consumption, let's say in the last 60 years or so. But yeah, we haven't, I don't know, maybe some of the techniques that they're using, they could even detect oil-based hydrocarbons. That's cool. That's something I haven't thought about. Good suggestion.
Here's another thoughtful technology question. Can you make ultra micro landers to use in streams, ponds, or rivers that we could use to monitor environmental changes all over in aquatic areas, fresh, salt, or brackish?
Yeah, we sure can. We actually already have some little landers just yesterday, we retrieved one that we call, or we don't call it, the manufacturer, good friend at Analytical Instrument Systems. It's called the WellStat, and it's only about the size of this here water bottle. And yeah, we just put it on a little shoebox sized lander made out of an aluminum frame, and we stuck it at the bottom of Lake Okeechobee so it can monitor for 1 month straight. Now, to get all the other bells and whistles, right, where you can actually detect nutrient fluxes and all that stuff, it's a little more complicated. But we do have some ideas for that. And I think the Lake Okeechobee stuff is really promising. I can't wait to look at the data that we just got back yesterday. But overall, yeah, the answer to that question is yes.
Okay, so are there indicators, or are there indications, sorry, that the blue holes you are studying have similar characteristics to blue holes off Mexico or in the Bahamas? And you know there's some pretty famous deep blue holes in some of those areas.
I think yes. You know, Nastasia, the lead author on this paper, she just did all that research, right? So to publish that paper, every time you publish a scientific paper, you gotta do an insane amount of research. It's actually too much. It's too difficult to publish a scientific paper these days because you have to provide hundreds and hundreds of references. But anyways, I digress. These blue holes, I feel like they formed the same way, some of them. And I wouldn't be surprised if many of them displayed some of the similar characteristics. I know that the one in China, or the one or two in China, do indeed display the redox stratification. So they do have the sulfidic bottom. And then they also display significant methane concentrations in there. So those researchers were looking at different, different chemistry or different analytes, but overall, yeah, some of the data is actually looking pretty similar. Now, with regards to the flow, I don't think I've seen any evidence of documentation of flows, especially this far offshore. There has been some flow documented near shore in the Gulf of Mexico, and some blue holes. I don't know if they're called blue holes. They're called just, I don't know, offshore springs, I guess, at that point. But yeah, there has been some flow demonstrated within a couple miles of shore.
So here's one from another one of our colleagues here, another professor here, Andia. And she says, excellent presentation. In terms of primary production, what are the major similarities and differences between blue holes and estuaries, if any? So it's pretty open-ended.
Yeah, and yeah, that's a tough question. Wait till it's your turn.
Yeah, exactly. Yeah, you'll have one first. But I let me just have a little spin on that. Do you ever see, because actually about 10 years before you got out there, your colleague Cutler he almost put another proposal in, and he was looking for somebody to go down there and do the algae with him. And so I was really excited because I really wanted to go out there. He never wrote the proposal, so I never got to go out. But he was telling me there are some that have these large quantities of deep water macroalgae. And that could be quite a story there. And again, it ties into nutrients and light and so on. You ever see that? Is there any signs of productivity out there?
Yeah, so I know folks from our team did take samples to look at phytoplankton community composition, but I haven't seen those results yet. So I don't know yet. And as far as, I guess one thing that we do know from the biological surveys around the rim is that there are seasonal differences in the seagrass around the rim. And so are those due to just generic sunlight differences just from that time of year and just warmth, temperature differences, or is it related to potential nutrient dynamics regarding the blue holes themselves? And so that's really the only thought I have on that one, Andia.
So we're kind of running out of time, but let me give you a couple more if we can. How will more blue holes be found? So you want to get a new one named after you. How are they? First of all, are they still being found? I mean, are they?
I think so.
Have they all been found, or
I doubt it.
How would one go out and look for them?
The ocean's pretty darn unexplored. So I mean, yeah, you can putter around with a bottom directed sonar. There's the technical term for them that's escaping me right now, but you can definitely chug along and look for them in like a lawnmower pattern, but it is very much resource intensive. So it's expensive to do that, right? You have to have somebody out there. So if you're really trying to look for these blue holes, which it may make sense to do from a financial, from a scientific, it may be justified from an ecological perspective, right? Maybe once we demonstrate how important they are, whether they're important or not, I think they are. But, you know, so maybe it makes sense even to have an autonomous vessel out there with a bottom mounted sonar looking for these blue holes. I think there are also, you can use satellites potentially to look for micro differences in temperature and salinity even, or just water color too. So or sea surface height and water color, I think could also be used, but it's all about pixel size and resolution.
Okay, so let's do one more. This is from Susan Allen, and Susan worked here a number of years ago, and it's great to see that she's here and she's asking a question. Now, I know you're not a biologist, but are there any benthic invertebrates in the blue holes or just microbes? If any benthic inverts, do they add to the puzzle of chemical fluxes? So good question.
A benthic invert, I think of octopuses, and we're talking snails, we're talking anything without a backbone, right? That's what we're talking about with the benthic invertebrate.
Yeah.
I mean, yeah, this is
Yeah.
you know, not something I think about every day. So
Yeah, I prefaced by saying I know you're not a biologist, but yes.
we saw a snail or two in green banana, and below that sulfide layer in amberjack, it's dead to our knowledge. There's nothing living. And it makes sense because there's no source of, you need an electron donor, an electron acceptor. If everything's just purely sulfidic, there's no, you know, yin to your yang. And so below the blue hole, below that sulfide layer, I don't think so. Above the layer, there is stuff stuck to the walls. So there are lots of sponges and whatnot that are stuck to the blue hole walls. And we do have a lot of video of that. And, you know, one day we will process those results, but there's obviously a huge backlog of data when we're doing this kind of work. So yeah, I don't know. I don't have a great answer.
Okay, well, I think that, you know, our time is pretty much up. I think that everybody knows that your email and your website is pretty available. Those they got to do is just Google Beckler, or just Jordon will do it. Jordon at Harbor Branch should get you right to him. And I don't want to steal your thunder, Jordon, but I know this other project that you alluded to, the Halo project, is probably just begging for a lecture next year. So I guess think about it.
Sweet. All right.
We hope to have you come back. You really generated quite a lot of good comments.
Well, thanks everybody. Yeah, I'm
very
just
exciting.
Yeah, happy to share this stuff. It's obviously exciting for me to share this info.
Yeah, and for everybody out there, again, we have a lecture coming up. Again, we're going to kind of continue with this idea of ocean health and ocean technology. So
Thanks,
see you
Dennis.
then. Okay, thanks so much.
Appreciate you hosting. Thank you so much.
Okay. Absolutely. Goodbye to everybody.
Bye.